Welcome to Azad Bioacademy. Today I will talk about the mechanism of insulin release. That means how insulin release from the beta cell. You know, the beta cell of pancreas is the factory of insulin. And the first question may come in your mind, that is, okay, beta cell will release insulin, but how it will get message? Now I have to release insulin. So two questions. One is how will get a message, or what would be the information based on it, the pancreatic beta cell will release insulin. And the second question, how it will release insulin. Okay, so firstly, the title is Mechanism of Insulin Release. Okay, so firstly, I will try to give you some basic, and then I will go to the details. So if you think this is the pancreatic beta cell, and this is the beta cell, and here is the insulin container, that means insulin vesicle, or you can say insulin granules. So if you take food, then the unit form of the carbohydrate, that will in the glucose, that will enter into the blood, and through blood it will reach to the pancreas beta cell, and it will just give signal to release the insulin. Another one is when you take food, your gut, your internal part of the GI tract will release particular type of peptide, that is incretin. And that incretins are basically GLP-1, GIP, like that. That means glucagon-like peptide and glucose-dependent insulinotropic polypeptide. These are the incretins. When you will take food, it will produce. Your gut will produce that. Your GI tract will produce that. And actually, carbohydrate is the main food to produce more incretin. Okay, and that incretin will reach to the blood and finally reach to the pancreas to release insulin. So this is the basic idea. Two information or two signal will reach to the pancreas for release. So now come to the details, how it will release. So if I just try to draw a pancreatic beta cell for my easy explanation, I will indicate the very large beta cell. So if you think this is the beta cell, this is the beta cell of pancreas. Okay. So here the surface membrane of the beta cell will carry a particular type of channel, transporter channel. Why I say this is the particular type? Because this channel is insulin independent channel. The transporter channel is called GLUT2. That means insulin independent glucose transporter channel 2. Without help of insulin, through this channel, glucose will enter from the bloodstream to your beta cell. Okay, so when you will take glucose or carbohydrate, then in your blood you will carry excessive sugar, and that sugar will enter into the beta cell. So suppose these are the sugar, and now sugar is in your inside of the beta cell without help of insulin. So through this GLUT2 channel, and this GLUT2 channel is present in the membrane of the beta cell. Okay, and then what will happen? That glucose will take part in the metabolic, it will go through the metabolic pathway. So before mitochondria, if you just think this is your mitochondria, before reaching to the mitochondria. Here, glucokinase, and then finally acetyl-CoA and then pyruvic acid, and then it will enter into the mitochondria, and then finally it will produce ATP. Okay, that means from the breakdown of glucose within your mitochondria, it will produce ATP. So when excessive glucose will be in your blood, and that will enter into the beta cell, and that beta cell will produce more ATP because more glucose will enter because in your blood excess glucose is available. So when your ATP production will increase within your cell, beta cell, ADP is comparatively low because ATP production is high. Okay, so what will happen? The ATP and ADP ratio will be more. That means your ATP is higher than ADP. You know, by the mitochondria, ADP can turn into ATP, and overall, through the metabolic process of the glucose, it will produce ATP. So excessive ATP change the ratio, and it will carry, now your beta cell will carry excess ATP. So then what will happen? That excess ATP what will you do to release the insulin? So I will just draw a special type of channel. This is on a special type of channel. Here is two things. This outer layer, that is, you can consider this is a channel, and inner part is carrying a channel as well as the inner part. Okay, this channel has two subunits. One is in the black color. You can think this is the KIR 6.2. That is potassium pore containing subunit, KRI 6.2. And the outer part is, you can consider this is sulfonyl urease subunit 1. Okay, so two subunit is there. One is KRI 6.2, another one is SUR1. Here SUR1 is the 4 subunit, and KRI also 4. So combinedly, the octamer of both two subunits will produce a particular channel. This is called ATP sensitive potassium channel. Shortly, you can say this is KATP. Okay, so what will happen? Actually, the KRI6 will carry the pore, and through this pore, the potassium will exit. So if you think these are the potassium. So normally inside the cell, potassium is very rich, and before entry of glucose, the production of ATP, basically the SUR subunit has two binding domain. One is a nucleotidic binding domain 1, another one is nucleotide binding domain 2. What does it mean? The ATP, ADP, these are nucleotide. That nucleotide will bind over there. The binding domain of the SUR1, that is sulfonylurease 1 subunit. It has the nucleotide binding domain. Okay, so in the resting condition, ADP will be there. That is ADP. And during that period, your potassium channel is completely open. Okay, potassium channel is open. And what will happen? In normal state, potassium will exit and it will produce hyperpolarization. So normally in the action potential, exit of potassium will create repolarization. But when the potassium will exit continuously, potassium efflux continuously, then it will produce hyperpolarization current. That means the below of resting potential. Okay, so in normal condition, no glucose entry in sufficient amount, and the beta cell is just working, and the membrane will produce hyperpolarization current through the ATP sensitive potassium channel. Okay, so now what will happen after entering the glucose into the beta cell? Before going to details, just for the easy understanding, I will draw here insulin vesicle. This is an insulin vesicle. You can consider this is the insulin vesicle, and it will carry your insulin. So I'm trying to draw 6 insulin, and here the center part is your zinc. So zinc will just carry 6 insulin within a vesicle. Okay, so hexamer of insulin will be available as a hexamer within the insulin vesicle, or you can say secretory vesicle, or simply you can say insulin granules. Okay, so before going to details, just I want to add something, another one, just rough understanding, that is, in your cell, that is, you can say this is the ribosome. It will produce pre-proinsulin. Okay, so 110 amino acid containing insulin is pre-proinsulin. So it will produce pre-proinsulin, and then it will go to the endoplasmic reticulum, and over there it will produce proinsulin, that is 86 amino acid containing. Okay, and then it will go to the Golgi apparatus, and it will produce 81 amino acid containing mature insulin, and it will produce insulin, and it will produce C-peptide. And by different vesicular protein and different types of uh, actually packing materials that will incorporate into the vesicle, and it will form insulin vesicle. And what will happen actually, why I come here, because in insulin vesicle, of course, the mature insulin will be present within the vesicle, but also equivalent amount of C-peptide is present. So if you think this is the insulin, so these are the C-peptide. These are the C-peptide. And during packing, during forming the vesicle, the C-peptide also will be available within the vesicle. Okay, so these are the insulin vesicle. Now come to the point. So when your beta cell will receive glucose through the GLUT2 channel, and it will just be metabolized, and it will produce ATP. Excess ATP will change the ratio, and excess ATP will bind where? The ADP or magnesium ADP or ATP, that is nucleotide binding domain. The main characteristics of that domain of the SUR1, that is sulfonylurease subunit, is the higher affinity towards the ATP than ADP. Okay, so when the VIP guessed, ATP will reach to that area, the ADP will out and ATP will bind over there. So ATP will bind ATP. Okay, then what will happen? Very simple. You can just think in your beta cell, within your beta cell, ATP is higher amount than ADP. So excess amount of ATP will just bind with the nucleotide binding domain, that is ATP binding site of the sulfonylurease subunit. Okay, then what will happen? It will change the conformation of the sulfonylurease subunit of the ATP sensitive potassium channel. And this is the tetramer of sulfonylurease subunit, and inside the tetramer KRI 6.2, which will carry the potassium pore. Okay, through this pore, potassium will exit. Already I said. So what will happen when it will make the conformational change of the sulfonylurease subunit, then it will shrinkage. The channel will shrinkage and potassium channel will close. Okay, your potassium channel will close. So no exit of potassium. So when in resting condition, when potassium exit, then it just cause hyperpolarization. But when now after binding the ATP, the potassium channel is already closed, then what will happen? Hyperpolarization will be inhibited. Then what will happen? Inside the cell, potassium is excess, the positive charge is excess. So what will happen? It will create a depolarization current. Okay, depolarization within the membrane. It will produce depolarization because hyperpolarization is stopped, and hyperpolarization is completely opposite of depolarization. Okay, so when depolarization current will be initiated by the closing of potassium channel after binding of ATP with the sulfonylurease subunit, then with the help of, now what will happen? That depolarization current will open a particular channel that is called voltage gated calcium channel. Okay, you can say this is the voltage gated calcium channel. Okay, that is the calcium channel. So if you think these are the calcium, here. So calcium will enter into the beta cell through this voltage gated calcium channel. Voltage gated calcium channel. The name reflects the meaning. There is depolarization current. When it will activate the voltage gated calcium channel, the depolarization, the intensity of current is changed, the voltage changes, and by the influence of this channel is the gate will open by the current voltage change. So depolarization current, when just reach to the voltage gated calcium channel, voltage gated calcium channel will open and calcium will enter into the cell. Okay, so these are the calcium. Then what will happen? That calcium will bind, just come up here, and it will bind on the membrane of the vesicle. And vesicle, where it will bind? Your vesicle will carry a particular protein that is called calcium binding protein, or you can say precisely synaptotagmin. synaptotacmin. So your calcium will bind with the synaptotacmin. And synaptotacmin is a special type of protein which is present on the membrane of the vesicle. When calcium will bind with the calcium binding protein, that is synaptotacmin, then it will change the conformation of the synaptotacmin. Synaptotacmin is tightly attached with the membrane. It will change the conformation. Then what will happen? It will produce a particular vesicular protein that is called snare protein. That is called SNARE, snare protein, and which is present on the membrane of the vesicle. So this is the snare protein B. If you just give some example, that is you can say BAMP2. Alternatively, the another name is synaptobrevin. That is synaptobrevin. Okay, so the BAMP2 or synaptobrevin, the alternative name, another name. These are the example of. This is the example of snare B protein. Okay, snare and B means the vascular, the membrane of the vascular. So snare protein will be activate. Then the rest motif will be just active on the surface of the membrane. Okay, so just I want to add something here. When it will release from the your Golgi apparatus, the vesicle, and it will be moving. That is, it will diffuse from one place to another. That is not static. Okay, some motor protein that can be kinesin and at the base, that is microtubule. Through this, they will just travel and it will stay near the membrane. Okay, so what will happen now? After binding the calcium, it will activate the further process, and then it will travel. It will move towards the membrane. Okay, so that means I wanted to say that it's before binding the calcium, it can travel by different factors, and now after binding, it will just reach to the membrane. How it will connect with the membrane, that is the question. So your membrane has also a particular snare protein. Your membrane also carry a particular snare protein. Suppose these are the snare protein which is present in the membrane of the cell, that is plasma membrane. Oh, so this snare protein is called snare T, and T is for target. So our main target is to just travel the vesicle to the membrane, and it will release. Okay, so this is the snare protein T, and here the particular snare protein T is the syntaxane, and you can say that is the SNAR snap. Sorry, okay, snap 25. These are the example of snap T. Okay, the snap T protein is present on the plasma membrane, and the snap B present is the membrane of the vesicle. Okay, already I said this is the, it will help to move towards the membrane. Then what will happen? Just I will consider here. So this is the vesicle, and it will carry the insulin, that is like that, and it will already, here is the snare protein B, that is synaptobrevin, or you can say BAMP2, and this is the syntaxine or SNAP25. Okay, and it will make a complex. The snare B and snare T will make a complex, and it will just make the vesicle in a right position, and it will just, you can say, it will be like that. Okay, and that snare protein will make the complex with the both snare T and V, and here another protein will work. That protein is called complexin protein. Complexin. That complexin protein is basically will uh, just determine the regulation. Actually, determine the timing. That is, this vesicle is mature or not. Premature vesicle will not be able to fuse on the membrane. That will not be allowed by the complexin. Also, that complexin will make a conformational change, and that will determine okay, this is the proper time to release. Okay, so this is the regulator on kind of. Okay, the snare protein T and B will make a complex, and that will now fuse the membrane. Okay, already attached, the membrane will be fused. Then what will happen? That fused form of uh, membrane, it will release the insulin. Okay, these are the insulin, and at the same time it will release the C-peptide. These are the C-peptide. Okay, so our main target is insulin, so we can consider these are the insulin. Oh, sorry, not no need to make any circle. So this is the insulin. And this insulin is released by the exocytosis. Exit from the cell, there is exocytosis by the fusion of the membrane of both vascular membrane as well as plasma membrane. And that will be made by the action of SNAP-T and V complex. Basically, syntaxin or SNAP25, and here is the VAMP2, or you can say this is the synaptobrevin. Okay, and here complexin will play an important role. Actually, here some other factors is working, that is REV, or you can say SAP160, different types of factors over there to make them more move towards the membrane, that you can say the motor protein, already I say kinesin, different types of protein are there. Okay, so now I will just sum up or summarize the things. When your blood will carry more sugar, the excess sugar will enter into your beta cell through the GLUT2 channel, that is glucose transporter channel 2. There is completely insulin independent, no need to insulin. Okay, that sugar will be metabolized within your cell, basically mitochondria, that will produce ATP. When it will produce excess ATP, the amount of ATP will be higher than ADP. That means ratio will change. That excess ATP will bind with the potassium channel, basically the ATP binding site of the sulfonylurea subunit. And then what will happen? When the sulfonylurea subunit will change its conformation, then it will make the shrinkage of that potassium channel, that is KRI 6.2, and then the potassium channel will close, and then what will happen? It will create a depolarization other than hyperpolarization. And by the help of depolarization current, the voltage gated calcium channel will open, and the calcium will enter from outside to inside, and that calcium will bind with the calcium binding protein, that is synaptotagmin, and then it will make the conformational change of the membrane, and that will make the rise of snare V protein, that is VAMP2, or you can say that is synaptobrevin, and that then in between its movement will just after binding of calcium, it will move towards the membrane. Also, before binding of calcium, some factors can work. Okay, then I will say the another snare T, that is targeted snare protein, is present on the plasma membrane, that can be said the the syntaxin or SNAP25, and that will make a complex between the synaptic the SNAP T and SNAP B, and here complex gene will work, and it will make a fusion of the both membrane, and it will release the insulin by the process of exocytosis. This is the insulin release mechanism by the glucose. Okay, and here at the time the C peptide will release, and you know the C peptide is basically work as like a marker of the insulin. Your body is producing more insulin or not. If you test C peptide in your blood, it will give you a message, because at a time same number of C peptide just released from the vesicle. Okay, so this is all about the glucose induced insulin release. Okay, so another point already I just initially I said, that is the incretin. Okay, so now come to the second point. In your membrane, a particular type of a receptor, that is incretin receptor. If you consider there is the GLP or GIP, so if you think the GIP, so GIP receptor is present, and if you think this is the GLP, so the GLP receptor also present on the surface of the membrane of the beta cell. Okay, so suppose I will say this is your beta cell receptor, that is incretin receptor. Incretin receptor, I will just give you, this is GLP-1 receptor, and GLP receptor on, suppose you can think this is the GLP. Okay, and GLP will bind here, and it will activate the receptor and active the. I try to draw the receptor as like a snake, that is serpentine receptor. This is 7-fast receptor, that is called G protein coupled receptor. So here, well, G protein will be there, and that is, you know, that is alpha beta gamma subunit. So this is the G protein. So when your GLP bind with the GTP, G protein coupled receptor, that is GLP receptor, then the G protein, this G protein type is called GS, that is stimulatory G protein. That means when GS will be activated, G protein, stimulatory G protein, then activated G protein will activate the guanylyl cyclase, which is present in the membrane, that is, sorry, adenyl cyclase. I'm so sorry. This is the adenyl cyclase. So when adenyl cyclase will be activated by the alpha subunit of activated G protein, then what will happen? It will produce cyclic AMP from the ATP. That means ATP will turn into cyclic AMP by the help of adenyl cyclase. Okay, and that cyclic AMP will activate the PKA, that is protein kinase A. You know, the PKA is the cyclic AMP dependent kinase. So PKA, protein kinase A, that protein kinase A will be activated by the second messenger cyclic AMP. Okay, now what will happen? That's PKA, protein kinase A. So you know the function of kinase enzyme, that PKA will just phosphorylate the voltage gated calcium channel. Then what will happen? It will open the voltage gated calcium channel. So then what will happen? The calcium will enter. Calcium will enter, more calcium will enter. You can say the PKA will phosphorylate and it will be activated more the voltage gated calcium channel, and the calcium will enter. So already you know the voltage gated, so voltage is needed, but when PKA will bind, it will just facilitate to enter more calcium from the outside to inside. So calcium will enter more, and that calcium will work as I mentioned earlier. Okay, so PKA is one of the major kinase enzyme which will be produced by the help of GLP binding to the membrane of the beta cell. Okay, and then another function, that PKA is directly activate to enter more glucose through the GLUT2 channel. Okay, so PKA also work on GLUT2 channel, and that GLUT2 channel will just receive more glucose from outside to inside. Okay, so this is another function of PKA. And here PKA also will help to translocate the vesicle from the inside to membrane. That means from the insulin release, the PK has a strong role because of different types of proteins are there, and some protein are inhibitory, and that inhibitory protein, PCB, that protein will be phosphorylated by PK, and you can say the RAB protein, and we can say SP160 will be more active to translocate the vesicle towards the membrane. Okay, so that means PKA will also facilitate to the release of to release the insulin. Okay, by the different function. That is, one is directly phosphorylate the voltage-gated calcium channel, and it will facilitate to increase the calcium entry, and also it will increase the entry of glucose, and also it will play an important role in the muscular changes during the insulin release. Okay, so in two way already I try to cover. One is through the glucose entry, that is glucose induced insulin release, and another one is GLP GIP, that is incretin induced insulin release. Okay, so in both way your body, your beta cell is just releasing insulin. Okay, not producing, that is releasing insulin. So if you think about the almighty creator, that is, when you are just taking some food, your gut just send the message to the pancreas. Okay, just get ready to release the insulin, because the people already get some food. Okay, and that will be digested from your within your GI tract, and then it will make a unit form, and it will absorb, that is sugar, and that will absorb into the systemic circulation, and that will reach to the GLUT2 channel. Okay, so it will take time. So in two ways your body is ready to release insulin when it is needed. Okay, so through the humoral pathway, that is, your blood carry the sugar, and that sugar will manage insulin. And another one is your incretin come from your GI tract, and it is just directly participating the insulin release. So this is all about insulin release. So if you have any question, then you can ask in the comment box or subscribe my channel.